كل شيء تمام صديقي.
Okay, welcome all and good evening. We are having today research, which is a big research in the field of Parkinson's disease. Before we start talking about the research, I would like to give a small brief introduction about Parkinson, the disease, the pathophysiology, and the motor physiology. If we had a coronal section from a human brain, we have know at first that we have some colored points in the brain. They're not colored in the reality, but they are dark. Those spots are groups of cell nuclei, so we call them nuclei, which is a group of cell bodies in the central nervous system. We have the cerebral cortex, the thalamus, the subthalamic nucleus here, and the putamen. And we have here the substantia nigra, which is the most important part in the Parkinson's disease. We have to know that every motor function is initiated in the cortex.
Like?
Yes?
Substantia nigra. بتعرف شو nigra?
Nigra is black.
Thank you.
Yes. And yes, it is a little bit darker from the other parts.
Exactly. عشان هيك اسمها. يلا go ahead.
Okay. Every motor function is initiated in the cortex, and from the cortex by neurons it goes to the putamen, and from the putamen to the globus pallidus, then to the thalamus, and then it goes back to different parts of the cortex, which is connected with the spinal cord to initiate movement. This pathway is called the direct pathway of motor function. Also, we have another pathway everything clear?
Yes.
Yes.
Yeah.
I had a problem in the computer. Okay. Sorry for that. We have also another pathway, which is also initiated from the cortex, and then it goes to the putamen. But the difference here is that it goes from the putamen to the subthalamic nucleus. From the subthalamic nucleus, it goes back to the globus pallidus, and then to the thalamus, and then to the cortex, and from the cortex to the spinal cord. And this is called the indirect pathway. The difference here is that the direct pathway is an excitatory pathway, so it excites the muscles. And the indirect pathway is an inhibitory pathway. It inhibits the muscles. Why do you think we need inhibitory pathways and excitatory pathways for the same action? Who have an answer?
يلا لين أول
Okay.
ضرب لين تفضلي.
To maintain homeostasis.
I don't think that muscles have a big role in homeostasis.
يعني homeostasis كلمة كتير واسعة.
Yes.
فبدي أفترض إنك أنت عم بتقربي من الجواب الصح. نسمع رشا.
according للphysiological conditions اللي بمر فيها الجسم، حسب إذا هو بحتاج حسب إيش مثلا إذا هو infected أو مثلا فيه إشي معين بحتاج إنه يكون inhibited أو عكس.
تمام، بس خليني أوضح لكم السؤال اللي سأله ليث. هو ليث عم بيسأل عن العضلات تحديدا، بيقول لك في
Yes, muscles.
العضلات في عندنا excitatory pathway لإلها وفي inhibitory pathway للعضلات. شو رأيك يا مجد؟
لأنه مثلا إذا بدي أعمل adduction بدي أعمل inhibition العضلات بتعمل abduction.
Yes, of course. I think that's the best answer. When we want to make flexion of the arm, we excite the biceps and inhibit the triceps, and so on in the all muscles. Also in the brain, mainly in the substantia nigra, we have other neurons that synapse with the direct pathway and the indirect pathway. Those neurons are called dopaminergic neurons, which contain dopamine as a neurotransmitter. And what happens in Parkinson's disease is that those dopaminergic neurons go in neurodegeneration. So they are dying. The role of the dopaminergic neurons that they control the direct pathway and the indirect pathway.
So when we have death in those neurons, we have the motor movement disorders that are symptoms of Parkinson's disease, like tremors, يعني زي ارتعاش في الإيدين, rigid limbs, stop posture, slow movement, inability to move, and also some non-motor disorders like GI disorders, heart rate variability, anxiety, depression, and so on. You can see here that the main hallmark of Parkinson
is that it is a highly progressive disease over time. So we have increase in the symptoms and increase in the disease over the time. If we had those dead neurons under the microscope, the dopaminergic neurons under the microscope, you can see that we have some dark spots in the cells. Those dark spots are called Lewy bodies. Those Lewy bodies are contained, or they contain alpha-synuclein protein. If you want to talk about alpha-synuclein, it's a protein that is mainly expressed in the brain and highly expressed in dopaminergic neurons. It doesn't have a definite specific function till now, but it's thought to regulate the neurotransmission of dopamine. If we had a normal alpha-synuclein, by some reasons, it will be misfolded, like insecticides, like toxins or oxidative stress in the mitochondria. We will have misfolded alpha-synuclein. These misfolded alpha-synucleins aggregate to form oligomers. Those aggregation can more aggregate to form fibrils. Those fibrils together can form Lewy bodies that are the darkest spots in the dead neurons. In 2003, a scientist that's called Heiko Prak, this is not the research of interest today, but he did research that he have took some biopsies from the brains and the nervous system of Parkinson's disease patients. And what he have noticed that if you have alpha-synuclein aggregates or misfolded alpha-synuclein in the higher parts of the brain, you will significantly have those aggregates in the lower parts, like enteric nervous system or the peripheral olfactory nervous system, and so on. If you have in the neocortex, you will have in the medulla oblongata and peripheral nervous systems. He concluded and hypothesized from that alpha-synuclein aggregation may not start in the brain. It may start in the gastrointestinal system. And what supports that hypothesis that a research that was conducted for 30 years for patients who had a vagotomy, which is a surgery in which part of the vagal nerve, you can see the vagal nerve in the orange color, which have a course from the brain to the GI system. Those patients who had vagotomy had a lower probability for having Parkinson's disease. How could alpha-synuclein move from the gastrointestinal system to the nervous system? Those aggregated alpha-synucleins can, like in a prion-like manner. A prion is a misfolded protein. Do you know what's a prion? We have taken that in the biochemistry course. Does anybody know?
Yes, it's a misfolded protein that is infectious to other proteins.
Yeah, that's right. Prion is a protein, as Omari said, are misfolded proteins that can infect other correctly folded proteins, so they become misfolded. So those proteins act like templates for other proteins. This aggregated or misfolded alpha-synucleins can move from a donor neuron in the enteric nervous system to other recipient neurons till they reach the brain by endocytosis or direct penetration or other types of movement. And now we have a question that what causes cell death? When a cell has aggregated alpha-synuclein or Lewy bodies, it's a foreign body. So that activates the microglia or the macrophages of the central nervous system to be activated. Those activated microglia, or let's start with the resting microglia, are highly branched. When they are activated, they become a little bit branched and more rounded with shorter branches. And what happens in the case of Parkinson's disease, those resting microglia are activated to the M1 phase, which is the pro-inflammatory microglia. Those pro-inflammatory microglia can induce neurotoxic function by releasing some cytokines like interleukin-6, TNF-alpha. Those are the most important for us. That can induce neurotoxic function and cell death. Everything is clear for now?
أنا عندي سؤال من شي درستكم إياه. المايكروغليا شو هي؟
Should I answer or from the...
الباقيين خلينا نشوف. أنت قرأتها أكيد. دانيا.
Kind of like the protectors or like
صوتك مش واضح ماما، صوتك مش واضح.
Can you hear me? Hello?
أيوة.
Okay. They're kind of like macrophages.
Kind of like? ليش kind of like؟ ليش kind of؟ احكي بوضوح، احكي عن وكوني واثقة من جوابك.
Yeah, they are.
تتذكروا لما حكينا عن الماكروفاج system؟
Yes.
They're part of the macrophage system, صح؟
Yes.
رائع، تمام.
They have different names depending on the area they're in. Basically.
صح. في حدا عنده إضافة؟ الناس رافعين إيدها ولا you agree؟ رشا لساتك رافعة إيدك، بدك تحكي شيء؟
عشان إنه كمان هي أصلا موجودة عشان البرين بلاد بارير ما بسمح لأي إشي زيادة يفوت، فبس عشان هيك موجودة المايكروغليا.
يعني مش بس عشان هيك، هي بتساعد وفي خلايا مخصوص شو اسمه للبي بي بي. حدا بتذكر مين الخلايا اللي بتعمل البرين بارير؟ فتحتي على حالك باب. يا عمر يحكي لي.
اللي كان إلها زي إيدين صح؟
شوف
هو أنا كنت بدي أسأل سؤال، بس بتذكر إنهم كانوا الاندوثيليوم كان صح؟ كان 3 لايرز حسب ما بتذكر. الأولى هي الاندوثيليوم، بصراحة نسيت الباقي، يمكن كان في نسيت الباقي صراحة.
مؤيد حسام. استروسايتس الخلايا النجمية، تذكروها؟
ايوه يس. بس عندي سؤال بالله لليث.
يس.
بتقدر تشرح لي اكثر شو بالضبط يعني نيوروتوكسيك فانكشن؟ السيتوكاينز شو راح تاثر؟
Yes, cytokines can fire up the neuroinflammation response. So the more microglia become T cells enter the tissue of interest. Neurotoxic functions induce the cell death.
اوكي تمام.
ثاني في شيء تمام كمان ولا خلاص؟
كل شيء تمام.
تمام. طيب هلا بدي اياكم شايفين هاي الصورة كتير مهمة. يعني سواء عم نحكي عن المايكروجليا او عم نحكي عن any other macrophage صراحة او macrophage like cell. طلعوا لي على هاي موضوع ال M1 وال M2. ال M1 pro-inflammatory، ال M2 anti-inflammatory microglia. هذا الحكي بنلاقيه في الانفلاماتوري situations. هلا عم نلاقيه كثير في انواع من الكانسرز. دائما بس يكون في ال M2 الريشيو تبعتها اعلى من ال M1. عم بيكون الريسيرش عم بيحكي لنا انه this is a better niche for recovery، يعني انه هاي المنطقة مثلا نفترض كانسر، بتروح هاي الخلايا الماكروفيج وفي عندها شوية ريزيستنس حتى للكانسر او مقاومة له، حتى في يعني مش بس في الانفلاماتوري، عم بيكونوا في الكانسرز. فهذه السويتش او الانتقال ما بين الام 2 والام 1 this is very very important. كل ما كان في عندنا ام 1 عم تتحول لام 2 اكثر، فهذا عم بيكون more protective، في حماية للتشو اللي موجود.
tissue repair function.
يس. جواد يس.
بس دكتورة بدي أسألك سؤال. الام 1 والام 2 للماكروفيجز نوعا ما جديد مش هيك؟ لسه يعني ريسنت ديسكفري.
يس.
في برضه سمعت عن المونوسايت برضه لهم سبسيتس. في برضه هذا حكي عنه يعني ولا لسه under investigation؟
هلا شوف، ما هي المونوسايت لا تنسى انه المونوسايت في البلاد. بدك تورجينا؟ ليه؟
No, I don't have something for now.
تذكر انه المونوسايتس هن هذا جبت لكم سؤال في الامتحان صح؟
هي اللي بتصير ماكروفيجز بعرف.
صح؟
بس ممكن
same idea. يس.
ممكن السبسيت معينة يكون لها مثلا affinity معينة مثلا للام 1 او ام 2.
يعني انا ما قرأت بس ممكن، يعني بفترض انه جدا ممكن، لانه اصلا شو اللي عم بيعمل differentiation لهاي او لهاي؟ اكيد الفاكتورز اللي around it.
شكرا دكتورة.
سجا, do you have something to add?
I actually have a question on something that was mentioned in the article, and as well, Laith also mentioned it. He said, and in the article, they said that patients that underwent vagotomy also had a reduced risk for Parkinson's disease. And
Yes.
I didn't understand the correlation between the two, to be honest.
Yes, I can. Okay. In the stomach, we have some cells that are called EECs, or enteroendocrine cells. Those cells can have an endocrine function, and they have a neurofunction, so they are connected to the enteric nervous system. When we have a toxin or anything that can induce alpha-synuclein aggregation in the stomach, this alpha-synuclein can be transported to those EECs. Those cells are connected to the nervous system, so the alpha-synuclein will move from those cells to the nervous system or enteric nerves, and then they will go through the vagal nerve to the brain. Is that clear?
Not quite, to be honest. I still.
Yes. You can ask the question.
يعني the surgery نفسها, like what would it affect? Like, why would they be at a lower risk of Parkinson's?
Because we have because we have cut a part of the vagal nerve, so we had decreased the probability for alpha-synucleins to move from the stomach to the brain.
Oh, okay. Yeah. Yeah.
Yes,
okay,
that's
that
the
makes
case.
Sense. Thank you.
You're welcome. Anyone have a question, or should we continue? Okay. So this is a diagram that can summarize what we have said for now, that alpha-synuclein aggregation can induce microglia activation and can induce Parkinson's disease. Okay. We have something special in the GI system of the humans that have taken the attention of the scientists in that field, which is the microbiome. Does anyone know what is the microbiome?
يلا اختار، بدك عمري ولا جواد ولا مجد ولا سجا أو رشا فيدانيا؟ ها في ناس عم تحمس.
The problem that I can't see who's raising his hand.
اسمع ما سمعنا من ليان. يلا يا ليان.
مرحبا.
أهلا.
أنا أول مرة أحضر معكم عشان هيك. الميكروبيوم هو كل الميكروبس اللي بتعيش بجسمنا، سواء كانت بكتيريا أو فيروسات أو any other things، but إنه بتكون الجسم مش عم يعمل ضدها immune reaction، بتكون يا مفيدة يا هي بتستفيد.
Yes, that's absolutely right. Thank you. The microbiome, which is the group of organisms that can live in the body, so the body doesn't make any antigens for their presence. And here we can enter our research with the hypothesis of the research, which says, can gut microbiota regulate motor deficits and neuroinflammation in PD? What they hypothesized that this microbiota can have an effect on Parkinson's disease promotion. So we are going to test something at first. We are going to test the presence of microbiota on the 3 sides of Parkinson's disease, which are the motor dysfunction, which are the symptoms, alpha-synuclein aggregation, which is the pathogenesis, and neuroinflammation, which is the pathway that can regulate cell death in Parkinson's disease. To do that, they have 4 types of mice. We are going to use a mice model for doing that. 2 of those mice will be alpha-synuclein overexpressive. Why do we use alpha-synuclein overexpressive mice? Does anybody know or have an answer? Why do we use alpha-synuclein overexpressive type of mice in this trial?
يلا يا زن ما سمعنا صوتك.
I think to increase the chance of them having Parkinson's disease, because if they overexpress it, there is more chance that something defective happens to it. So there is more chance for Parkinson's disease to happen to the mice. I think that's the.
But that's really large
explanation.
Yeah, that's the answer. Absolutely. We use. Thank you. We use alpha-synuclein overexpressive mice, so those can simulate the type of people that are genetically can induce Parkinson's disease. And many research was done to prove that the alpha-synuclein overexpressive is the best model of mice to simulate motor dysfunction in humans. And we have now a wild type, which can simulate the people who don't have genetic probability to induce Parkinson's disease. And then after we have those types of mice, we made, or they made, another 4 subtypes of them. So we can now test the presence and absence of microbiota. We have ASO mice that are germ-free. They don't have microbiota. And also wild-type mice that are also germ-free. And we have overexpressive mice for alpha-synuclein that are specific pathogen-free. So they have microbiota, but they are specific pathogen-free. So we don't have other variables than the ones that we are going to measure. Also, we have wild type that are specific pathogen-free. Is everything clear for now? For the types of mice that we are using in the trial?
أنا عندي سؤال.
Yes.
ما فهمت شو يعني specific pathogen free.
Specific pathogen free, that those mice contain microbiome or contain bacteria, but scientists have removed the bacteria that can induce pathogens.
Pathogens in general ولا لها دخل بالباركنسون؟
We don't know for now, but every bacteria that can induce disease, they removed it from the microbiome.
Okay.
The microbiome or the relation of microbiome and the Parkinson disease is not induced by an infection of bacteria. We will talk about that later.
ليث عبادة عنده سؤال.
مرحبا.
أهلا. بس ممكن تعلي صوتك شوي.
كيف من وين جابوا germ free mice? Do they naturally have no microbiota ولا they removed it, the scientists?
They grow them in a very clean environment. So يعني من الولادة كانوا by a technique they can grow germ free.
Okay.
ليث أنا ما فرق بين ال SPF وال germ free.
Germ free, they don't have microbiota
at all.
At all.
Will SPF?
SPF have microbiota, but they are specific pathogen free.
Okay.
Did I clarify that?
يعني عندهم الجراثيم اللي موجودة نورمالي موجودة في
عندهم الميكروبيوم.
اوكي ولكن
selectively.
ما في شيء ممرض pathogenic صح؟
Yes.
يلا go.
So we don't have other variables of pathogens.
تمام.
We have a question now that how can we measure the motor function of the mice? There are some tests that scientists agreed that can be used to measure the motor function. We have the beam transversal test, which is a test. We have two ends and a beam in the middle. We put the mice on one end and let them go to the other end and measure the time to do that. Syn test is the same test of the beam transversal, but it's in the different way that we put the mice in the upper pole of the in the upper end of the pole and let them go to the bottom and measure the time to do that. The nasal adhesive removal is a test where we put something adhesive on the nose of the mice and measure the time that the mice take to remove that adhesive something. Hindlimb clasping reflex is a test in which we hold the mice from their tail upside down and measure that, and we have a specific scoring scale for the times the mice make a clasping. So touch the limbs together. And logically, all these tests, if the mice are have motor dysfunction, they will take more time to do that test and that test and that test and higher score in that test. What we wanted to measure at first, that the effect of presence of microbiota on motor dysfunction, which was our first test here. As you can see in the diagram, that the mice that are ASO and they have microbiota took more time to do the first test and the second test and the third test, and also had a higher score in the last test. And they are significantly higher from the ASO mice that are germ-free and significantly higher from the wild type. And also the same thing here and here and here. So we can conclude that the presence of microbiota can really affect the motor function of the mice. Everything clear for now? Or anybody have a question?
ما حدا عنده سؤال شكله ليت كمل.
Yes, okay.
اسمع بلا ارجع اعيدها بس. ارجع اشرح الدايجرام لانه شوي خربطت.
Okay. As you can see here, that the mice that are overexpressive of alpha-synuclein that have a high probability of the Parkinson disease, and they have microbiota, have taken more time to do the test. And that implies that they have higher motor dysfunction. And the same thing happened in the second test, third test, and the fourth test. The second thing that we are going to measure is the presence of... Yes.
احكي لنا الاستنتاج.
I said that. This data that you can see here imply that the presence of gut microbiota can promote the hallmark motor dysfunction in a preclinical model of Parkinson's disease, because the mice that had microbiota took a longer time in all the tests significantly. So they had a higher motor dysfunction.
تمام.
The second thing that we are going to measure is the effect of presence of microbiota on alpha-synuclein aggregation, which is the pathogenesis of the Parkinson's disease. What we have done here is that we used immunofluorescence microscopy, in which we use pink or red fluorescence antibodies to the alpha-synuclein aggregations, so that alpha-synuclein aggregations can appear in a pink color using antibodies that are specific for them. And you can see here that the SPF ASO, which contain microbiota, had a really higher rate of alpha-synuclein aggregation in comparison of the germ-free. So you can see that the pink dots are alpha-synuclein aggregates. In comparison of that, we have no alpha-synuclein aggregates here, which are germ-free. And the same thing happened in the substantia nigra, which is another part. Those are in the codopotamin pathway. Also, they have, wait a minute. Okay. Also, to prove the results, they did Western blot for the soluble, which is the normally folded alpha-synuclein, and insoluble, which is the misfolded alpha-synuclein. We did Western blot, and you can see that the specific pathogen-free type of mice had a lower concentration of soluble, which is normally folded alpha-synuclein in comparison of the germ-free. And if we look at the insoluble, we can see that the specific pathogen-free has a higher concentration of insoluble, which is alpha-synuclein aggregations, in comparison to the germ-free. And this is a diagram for the ratio of insoluble to soluble, and you can see that it's really high in the specific pathogen-free in comparison to the germ-free. And these data suggest that the presence of microbiota can really promote alpha-synuclein aggregation in the mice. The third thing that we wanted to test is the effect of the presence of microbiota on neuroinflammation, which is the pathway that can control the cell death. We have used here a 3D construction technique, which is a technique of imaging for the cells in a 3D images. As you can see here, that the specific pathogen-free in the wild type have a highly activated microglia, and the same thing happened in the ASO type. And we can conclude that the presence of microbiota can really activate microglia in the comparison of the germ-free, as you can see here. We said in the previous slides that the activated microglia is more rounded with decreased branching. And that's what you can see in those images. Also, they have did a quantification of that by the diameter. You can see that ASO SPF has the highest diameter, which means that they are highly activated, and the lowest number of branching, and also the branching length. And the same thing in the substantia nigra. Also, to prove the results, they have done ELISA test to measure the concentration of TNF alpha, also the interleukin 6. Do you know what are these? We have talked about that in the previous slides. TNF alpha and also interleukin 6.
Yes, Majd has an answer.
Cytokines?
Yes, of course. They are pro-inflammatory cytokines. So we can say that the presence of those cytokines, which are TNF alpha and interleukin, in high concentrations, that means that we have an inflammation and also highly activated microglia. And you can see here that ASO, which contains the microbiota, have a high concentration of TNF and also a high concentration of the interleukin 6. Also the same thing in the midbrain, where we have high concentration of TNF alpha, also a high concentration of interleukin 6. What they did here, that they have taken another parts of the brain, which is the frontal cortex and the cerebellum. And you can see that there is no difference in the CB and no difference here in the TNF alpha, and also no difference in the diameter. On the other hand, we have difference in the frontal cortex with here you can see here that the ASO SPF high diameter and also have higher concentration of TNF alpha. We can conclude from that the inflammation happens in the parts that are important in the disease, or they have intervention with the disease or effect in that. And the whole conclusion of that gut microbes promote alpha-synuclein dependent activation of microglia within specific brain regions involved in the disease. Everything clear for now? Does anyone have a question?
عمري.
Yes.
هسا أنا هاي ما فهمت هالشغلة من أول. الباركنسون disease does it occur in specific spots in the brain?
Yes, we have to go back. Wait. we have said here that the main hallmark of Parkinson's disease is the degeneration of the dopaminergic neurons in the substantia nigra, which is this part of the brain.
Okay.
Where we have dopaminergic regions.
اوكي شو اللي قسنا وين ايش الاماكن اللي قسنا فيها ال
we have also dopaminergic neurons in the putamen, also in the caudate nucleus, where we have the caudoputamen pathway here. We have also dopaminergic neurons.
But did they measure the I forgot its name, the Negra, the Negra part? Yes.
Wait. Is this the slide? Yes, you can see that we have measured that in the caudoputamen. We have measured the substantia nigra here.
Yes.
Also, we have measured in the midbrain, which is involved in the disease, and also in the caudoputamen pathway.
Okay, تمام.
More questions?
شكله ما في. Go ahead.
Okay. If we want to think about this research in a therapeutic pathway, we have to be more specific in our research to know when to interfere with our drugs and what specifically to do, or what receptors should we make the drug for. So they made further research to know is this signaling from the microbiome to the brain and effect, is it prenatal or postnatal? So they had two types of mice, which are the SPF mice and the GF mice. What they did that they treated the SPF mice with an antibiotic cocktail treatment, so they became APX mice. So they are treated with antibiotics and became germ-free. And the germ-free mice were colonized by SPF microbiome, and they became XGF mice. So they now harbor microbiome, and the APX mice are now germ-free. And they tested for the different tests, as you can see here, the time to cross the beam. These are the motor function tests here. This is the first test, the second, the third, and the fourth. As you can see here that the XGF mice have the same results of the SPF mice. And the GF mice have also the same results of the APX mice. Also, that's the case in the second test and the fourth test, and also the third test and also the fourth test. If you can see here that the APX mice have less higher. This is less microglia activation than the XGF mice. So the XGF mice have the same results of the SPF mice, and the GF mice have the same results of the APX. And the conclusion is that the modulation of microglia during adulthood contributes to alpha-synuclein motor dysfunctions and neuroinflammation. And that suggests that the signaling between the gut and brain by the microbiota is still active in the adulthood. So we can interfere in the adulthood with our drugs to affect that pathway. Clear for now? Any questions?
No.
Go ahead.
Yes.
ما شكله حدا عنده أسئلة. يلا كمل.
Okay. Also, we wanted to be more specific, and we asked a question. Is the bacteria itself what regulate the neuroinflammation and motor dysfunction in PD? What arised this question is that a research which was conducted in 2015 has revealed that the gut bacteria modulate microglia activation during viral infection through the production of microbial metabolites, which are named short-chain fatty acids. Short-chain fatty acids are metabolites of bacteria. Those are examples of short-chain fatty acids: acetate, propanoate, and pyrrolate. Those are examples of short-chain fatty acids. A research in 2015 have revealed that they can regulate the infection in viral infections. They can regulate the inflammation in viral infections. And that was the research that arised this question. To make sure of that, they did further tests and tested the effect of the presence of short-chain fatty acids on motor dysfunction and also alpha-synuclein mediated neuroinflammation. And they get those results. You can see here that if we had SPF mice, also GF mice, and we have treated some GF mice with short-chain fatty acids. So we had GF mice. We have treated them with short-chain fatty acids, so they become CSFA. You can see that the results we get from the short-chain fatty acid treated are the same results of the SPF mice. Also, that's the case in the second test and the fourth test. If we also seen the neuroinflammation, you can see that if we had ABX mice, which are antibiotic treated, so they are now germ-free, and we had the XGF mice, which are now SPF, and compared that to the CSFA, we can get that the results are the same for those two and different from that. Also, it's the case for the substantia nigra part. And that really gives us a conclusion that what really promotes the alpha-synuclein aggregation and neuroinflammation are short-chain fatty acids, and they are not the bacteria itself, because they also treated some germ-free mice with heat-killed bacteria, and they gave the same results of the germ-free mice, as you can see here and here. And that really proves that the short-chain fatty acids are the sufficient molecules to promote alpha-synuclein mediated neuroinflammation. Any question for now till now?
ليث أنا عندي سؤال.
Yes.
يعني هن عم بيحكوا إنه الأنتيبيوتيك في هاي الحالة أو الأنتيبيوتيك treated mice would give you the same result as germ-free، وبالتالي they're suggesting إنه antibiotics ممكن تقلل من the incidence of PD.
That's not the case that what they suggested in the research, but they say that we can give a treatment which can interfere with the microbiota.
Did they suggest which type of antibiotic؟ ولا حكوا مثلا spectrum ولا على أي بكتيريا تحديدا تشتغل؟ gram positive أي شيء؟ ما حددوا.
We will have more experiments for the experiment now, which can give more idea about that.
بس القصد إنهم ما سألوا شو نوع الأنتيبيوتيك، شو نوع البكتيريا اللي
No, they say that we can use the test to make more therapeutics and etc.
أوكي. أتفضل عمري، إيش بدك تحكي؟
يعني أنا هسه اللي عم بفكر فيه إنه إحنا مش محتاجين إنه نعمل أنتيبيوتكس للميكروبيوتا نفسها، كونه إحنا وصلنا لقناعة نتيجة حاليا إنه شورت تشين فاتي أسيدز هي المين كوز. أنا هيك فهمت لحد الآن، هيك فهمي صح يا ليث؟
Yes, we can use some therapeutics that
to interfere with
different...
short chain fatty
Yes,
acids specifically,
that's the case.
without getting into
because I don't think it's a good
antibiotics.
idea to kill the bacteria.
Yes.
Because of course they have different roles from the short chain fatty acids.
بالضبط.
Yes.
They did further research. What they did now, we have a question. What do you think would happen if we colonized mice with Parkinson patients derived microbiome in comparison with healthy humans derived microbiome? So if we took germ-free mice and we colonized in part of them Parkinson microbiome, Parkinson derived microbiome, and we had the other half that we colonized with healthy humans, what do you think would happen if we did that?
سجد تفضلي.
So I think that the microbes that we get from the PD patients are going to impair the motor functions of the mice that we're gonna put the microbes in, relative to the healthy humans, the microbes taken from the healthy humans that don't have PD.
Yes, thank you. That's the case. What happened? If we did that, you can see here that the Parkinson disease microbiota have the impairment in the motor function, in comparison of healthy patient microbiota in most of the pairs. And this is compiled. The results are compiled. You can see that the motor dysfunction here is higher than the healthy microbiota. And what's really the case that Parkinson's disease patients have different microbiota composition from healthy patients. So what we can conclude, this finding in a preclinical mouse model supports the notion that PD microbiota contributes to the disease symptoms in genetically susceptible hosts. And we have a final conclusion of this research that can say, this is quoted from the research: our findings establish that the microbiota are required for the hallmark motor and GI dysfunction in a mouse model of Parkinson's disease via postnatal gut-brain signaling by microbial molecules, which are the short-chain fatty acids that impact neuroinflammation and alpha-synuclein aggregation. The importance of this research now, that we can use that we can interfere with the microbiota or the short-chain fatty acid concentrations to affect the neuroinflammation. What's interesting in the neuroinflammation, that we can slow the cell death. What we said in the previous slides, that Parkinson's disease is a highly progressive disease. So when we use the traditional drugs for Parkinson, like levodopa, carbidopa, and others, we need higher concentrations of those drugs, so they become ineffective. So if we can interfere with the disease by a neuroinflammation pathway and short-chain fatty acid concentrations, we can make the disease slowly progressive. So we can use a good concentration of drugs for the patients. And that was the end of the research. Anyone have a question for now?
في ناس عندهم أسئلة، بس أنا بدي أقترح عليك اقتراح.
Yes.
قبل ما نفكر في موضوع يعني لو إحنا researchers، وقبل ما أروح أفكر في موضوع الدراج to target مثلا هدول fatty acids or something, why don't we أول شيء assess which microbiota are associated with PD and which are associated with the healthy? وفي research كتير في هذا الموضوع.
I think that those were mentioned in the research, but it was highly detailed. I didn't want to explain that in the presentation.
في كتير كثير شغل بهذا اللاين إنه وات تايب اوف
Yes.
مايكروبيوتا، مش بس بهذا، في كثير على الموضوع و very recent articles كمان. هلا الإشي الثاني إنه يعني النكست ستيب إنه how can we affect هذا السويتش من خلينا نسميها الباد مايكروبيوتا اللي هي associated with disease لجود أو beneficial مايكروبيوتا. فيعني هاي من الأشياء اللي ممكن نفكر فيها. مش دائما بيكون الدراج هو الطريقة السليمة. تعرف شو الحديث؟ المعدة بيت الداء.
We can interfere in other ways other than drugs.
Yes.
Yes, I think that better.
كثير كمان حكي على هذا الحكي، يعني على هاي الطريقة إنه شو النيوتريشن له علاقة.
Yes, the nutrition have a relationship with that.
تمام. رشا كانت رافعة ايدها عندها contribution. تفضلي رشا.
مرحبا. بس ترجع شوي على السلايدين اللي قبل، أو لا ارجع هيك. لا وين ما كنت؟ أيوة هون. هلا بس بدي أسألك سؤال، بدي أفهم شغلة. لا ارجع على الشارت.
اوكي.
هلا أفهم من حكيك إنه الميكروبيوتا لما تكون مع شخص صار عنده باركنسون ديزيز تختلف عن إنسان، تختلف نوعية الأشياء اللي بتطلعها عن إنسان طبيعي.
Yes, when we have different concentration or presence of different types of bacteria, we will have absolutely different concentration of the metabolites of those bacteria.
إحنا علاقة بإيش إيش بيطلعوا كفاتي أسيدز، يعني بيختلف من طبيعة إيش بيطلع products.
Yes.
أوكي شكرا.
أهلا. More questions?
رشا راح أحط لكم هلا article، والجميع راح أحط لكم زي review article فيه شوية تفاصيل عن gut microbiota associated with Parkinson. إذا حبيتي اقرأي. سجا كمان، وبعدين جواد، وبعدين عمري. تفضل.
First of all, يعطيك العافية, Laith. It was an amazing and interesting presentation. One of my questions, I have two. The first one is, why didn't they mention the short fatty acid chains in the conclusion? Because didn't they present very convincing evidence that it's the short fatty acid chains that play a role rather than the actual microbiota?
It's not in the conclusion. It's part of the research, but it was in the end of the research. What they did that they treated some bacteria, some mice with heat-killed bacteria, and nothing happened. Also, they were affected by this research, which said that the short yes.
ليث هي قصدها عن الآخر experiment اللي هي لما جابوا من، أتوقع هيك، لأنه هذا السؤال كنت بدي أسألك إياه، إنه لما أخذوا مايكروبيوتا من PD patient and from a normal person، ليش ما صار في measurement للshort chain fatty acids؟
Yeah, that's the question.
I really don't know the aim of measuring the concentration of short chain fatty acids, because when we have different microbiota concentration, we will have different concentrations of short chain fatty acids. I think I have read that in the research, but I have to reread that to give an answer.
Yeah. Okay. Thank you. So that's the first question. And the second question, Yani, either you or Dr. Hanan can answer this. Excuse me. For the short fatty acid chains, is it, Yani, does your diet also play a role, or is it simply the short fatty acid chains that are produced or released from the microbiota? So, for example, does the type of food you eat make you more susceptible to Parkinson's disease, for example? Does your diet play a role?
The diet plays a role mainly in affecting the alpha-synuclein aggregation, which can start in the gut. But I think also the diet can affect the short-chain fatty acids concentration. What do you think, Dr.
Yeah, because I've read. Oh, okay, never mind.
كملي سجة كملي.
Yes.
Because I've heard that, well, not have heard, it's common knowledge that certain foods stimulate or encourage the production of the short fatty acid chain. So I was wondering, if you eat more of those foods, then you're more likely to develop aggregates, which eventually would cause the Parkinson's disease. And I'm not sure if this is specifically to Parkinson's disease or if it's related to Parkinson's disease or another disease, but I remember reading that they also have limitations to their diet and what they can eat. So I don't know if that's the reason, or if it's also Parkinson's disease, or I'm mixing it up with another disease.
I really don't know much about the effect of diet on Parkinson's disease.
زي ما قلت لكم هلا الافكت اوف دايت عم بتكون عن نيورو ديجنريتيف ديزيزز والنيورو انفلاماتوري ديزيزز، في a lot of studies عليها. ف يعني فيكم تفتحوا على سكولر جوجل ودوروا بس شو الهيتس اللي عم تطلع amazing work صراحة يعني. وإحنا لأنه بعالم الطب مش متعودين على هاي الأشياء البديلة، يعني usually اللي إحنا متعودين عليه symptoms بنروح بنعملها دواء وبنعالج المرض، وهذا اللي بنفهمه. هلا جسمنا بيشتغل بطريقة مختلفة. يعني المايكرو انفايرمنت اللي جوات جسمنا is affected بكتير أشياء، من ضمنها الفود، من ضمنها الميكروبيوتا الموجودة، من ضمنها كتير أشياء ممكن إن الواحد يعمل ريسيرش فيها. فيس سجن ممكن جدا اللي عم تحكي فيه. يس تفضل يا جواد.
بدي أسأل بس سؤالين بسيطات. الأول ليث، إحدى التجارب اللي أنت حكيت عنها إنهم حطوا microbiota from PD infected patients or PD symptomatic patients in
Yes.
mice. طب ليش ما جربوا مثلا إنهم يجيبوا healthy microbiota from a healthy person and infect a diseased mice؟ ممكن يكون لها أثر ثاني، بجوز يكون البروجريشن أبطأ من مايس ثاني عنده unhealthy microbiota مثلا، خلينا نحكي for lack of a better word يعني.
I think that's a good question, but I really don't know. If you want to think about that in a simple way, you will probably bring some germ-free mice and give one of them Parkinson's disease and one of them healthy microbiota. But why they didn't do what you said, I don't know really.
بس يعني ممكن تكون تجربة منيحة، ممكن تكون مفيدة يعني. شو رأيك
Yes, I
دكتورة؟
think that will be a good addition to the research.
يا أكيد، بس يعني بفترض إنهم عملوا هيك just to expedite، يعني بس لتسريع العملية. أوكي عمري.
أنا بس كنت بدي أحكي على السؤال رشا وسجا، اللي هي على قصة إنه الميكروبيوتا فيها فاريشن بين كل واحد والثاني بحسب اللايف ستايل تبعه. عشان هيك فعلا الدايت ممكن يكون لها أثر مباشر على الميكروبيوتا بإنها تكون مثلا secretes more short chain fatty acids. حبيت أحكي بس هاي المعلومة.
Thank you عمري، صح مظبوط.
أنا بدي أقرأ جملة من الريسيرش دورت عليها. كان هنا مكتوب what causes dysbiosis in PD patients. Dysbiosis يعني different microbiome. كان مكتوب one of the reasons reduced gastric motility or dietary habits. So I think that dietary habits really affect Parkinson's disease promotion.
يس تمام. إذا حدا حابب يجيب لنا معلومات كمان عنه، أو إذا بتحبوا I can upload an article. خليني هلا أحط لكم الارتيكل اللي حكيت لكم عنه اللي له علاقة بالميكروبيوتا، وممكن كمان كم من يوم أرجع أحط لكم كمان ارتيكل ثاني عن الدايت والبي دي. وزي ما قلت لكم مش بس البي دي، يعني كتير هلا في ريسيرش على الام اس مثلا، مش بس على الاي ال اس. كل هدول نيورو ديجنريتيف نيورو انفلاماتوري ديزيزز عم بيدوروا على الريليشن تبعتهم.
الزهايمر كمان
والالزهايمر يس.
بس في عندي إضافة أخيرة. الريسيرش كثير طويل وكثير معبى معلومات، فبعتقد لو ترجعوا تقرأوه مرة ثانية بعد ما شرحناه you can fill the gaps، لأني أنا اختصرت منه أشياء.
تمام. Thank you so much ليث، يعني it was really interesting.
You're more than welcome.
في حدا عنده أي إضافة أو أي سؤال لليث؟ طيب الله يعطيك ألف عافية. إحنا اتفقنا إنه بس دراسة نعمل every 2 weeks.